Chromosomes are the units of inheritance. Genes, which contain the instructions to produce RNA in proteins and cells, are found on chromosomes. Humans inherit 23 chromosomes from each parent. The first 22 pairs of chromosomes are completely homologous. The 23rd pair, or the sex chromosomes, are also homologous if the individual is a female and has 2 X chromosomes. If the individual is a male, however, only a small portion is homologous, so that the X and Y chromosomes will synapse during meiosis. During meiosis, independent assortment occurs. When the homologous chromosomes line up in metaphase 1, the orientation of the maternal and paternal chromosomes are random. This produces varied haploid gametes. If 2 genes are found on one chromosome, they are likely to both be found in the gamete. We say the genes are linked. But due to crossing over, genes on the same chromosome may become unlinked. The closer the genes are located on the chromosome, the more likely they are to still be found on one chromosome even after crossing over. Remember crossing over? Well, that can unlink genes. During crossing over, bits of maternal and paternal chromatids exchange places, and we get recombined chromatids. So the chromosomes that end up in each cell at the end of meiosis are different. 2 cells contain the recombined chromosomes that have some maternal genes and some paternal genes. This further increases the combinations of alleles offspring can receive. Non-disjunction occurs when homologous chromosomes, or sister chromatids, fail to separate during meiosis, resulting in an abnormal chromosome number. Non-disjunction may occur during meiosis 1 or meiosis 2. If non-disjunction occurs in humans during meiosis, this can result in different disorders. If it occurs in autosomes, the resulting fertilized egg will most likely not develop. However, offspring with an extra chromosome 21 can be viable, but have trisomy 21, also known as Down syndrome. In some cases, additional sex chromosomes are not as critical. Many genes necessary for development are found on the X chromosome, so at least one copy is required. But only one chromosome remains active in cells. Additional X chromosomes are inactivated. This means individuals with 1 or more than 2 X chromosomes are not uncommon. But some organisms naturally have additional chromosomes. Tardigrades have 3 sets, some flowers have 4 sets, cultivated strawberries have 8 sets, and sturgeon have up to 10 sets of chromosomes. They are also known as polyploids. For example, wild strawberries are diploid, but cultivated strawberries are octoploid. Do you remember that in female mammals, one X chromosome is inactivated so that only one gene from each chromosome is active? The inactivated X chromosome is known as a Barr body and can be observed microscopically. In cats, the gene for coat color is located on the X chromosome. In the embryonic development of female cats, one of the 2 X chromosomes is randomly inactivated in each cell, resulting in a tortoiseshell pattern if the cat has 2 different alleles for the coat color. Male cats, having only one X chromosome, do not exhibit a tortoiseshell coat color. Chromosomes are complex and can be damaged in the cell during DNA replication, mitosis, or when genes are copied by a process known as translation. Repair enzymes can repair the damage, but sometimes errors are made. An inversion is when a segment of DNA breaks at the phosphodiester bonds on both sides. The segment then randomly turns around and is reinserted, so that the strands of the segment are inverted compared to the rest of the DNA. This changes the nucleotide sequences for both strands of DNA in the region of the inversion. Translocation is when 2 chromosomes break and the broken parts are swapped. Again, this changes the nucleotide sequences on the chromosomes. However, if genes are exchanged in their entirety, they may still be functional in the new chromosome.